Hi, now that we finished the chapter on chemistry, I hope you have a greater appreciation in terms of molecules and how these molecules can bind together by covalent bonds. And there can be other characteristics such as ionic bonds in which the electrons are actually transferred between atoms, and even the properties of water, which allow different living organisms to live on this planet Earth, and even from a molecular level in terms of water acting as a solvent. One of the key things about these particular molecules that we looked at, particularly the macromolecules, is that within the course of evolution, three of them, mainly the proteins, the phospholipids, and the cholesterol, coalesce together to form what we now know as the plasma membrane. And it's the plasma membrane that is going to be unique and not unique. Plasma membrane is going to be found in all cells, whether they're prokaryotic cells, animal cells, plant cells, fungi cells, protists, etc. And it's the plasma membrane that will allow the internal environment, in other words, the cytosol, or the cytoplasm, as some of you might know, to have a unique chemical environment compared to that of the external environment. So the external environment can be one thing, but the internal environment could be something very unique. So the plasma membrane allowed the separation between the internal environment and the external environment in terms of cells. So that was one of the big jumps in terms of the course of evolution for living things. During the course of human endeavors and discoveries, some gentlemen basically came to look around in terms of what were some of the things that they discovered. Well, let's say there's Robert Hooke, there's Van Leeuwenhoek, and these guys came together, and not physically, but both of them essentially discovered that if they were to build this particular thing called a microscope, they were able to amplify or at least magnify things that they were not able to see prior. And in process, over the course of a few hundred years, this cell theory eventually came into being. And the gentlemen behind the cell theory, or three of them, Schleiden, Schwann, and Virchow. And rather than me explain about that, I think I have a really good little clip that I would like for you to watch in terms of the actual cell theory.
One of the great things about science is that when scientists make a discovery, it's not always in a prescribed manner, as in only in a laboratory under strict settings with white lab coats and all sorts of neat science gizmos that go beep. In reality, the events and people involved in some of the major scientific discoveries are as weird and varied as they get. My case in point, the weird history of the cell theory. There are three parts to the cell theory. 1. All organisms are composed of one or more cells. 2. The cell is the basic unit of structure and organization in organisms. And 3. All cells come from pre-existing cells. To be honest, this all sounds incredibly boring until you dig a little deeper into how the world of microscopic organisms and this theory came to be. It all started in the early 1600s in the Netherlands, where a spectacle maker named Zacharias Janssen is said to have come up with the first compound microscope, along with the first telescope. Both claims are often disputed, as apparently he wasn't the only bored guy with a ton of glass lenses to play with at the time. Despite this, the microscope soon became a hot item that every naturalist or scientist at the time wanted to play with, making it much like the iPad of its day. One such person was a fellow Dutchman by the name of Anton van Leewenhoek, who heard about these microscope doohickeys, and instead of going out and buying one, he decided to make his own. And it was a strange little contraption indeed, as it looked more like a tiny paddle the size of a sunglass lens. If he had stuck two together, it probably would have made a wicked set of sunglasses that you couldn't see much out of. Anywho, once Leewenhoek had his microscope ready, he went to town looking at anything and everything he could with them, including the gunk on his teeth. Yes, you heard right. He actually discovered bacteria by looking at dental scrapings, which, when you keep in mind that people didn't brush their teeth much, if at all, back then, he must have had a lovely bunch of bacteria to look at. When he wrote about his discovery, he didn't call them bacteria as we know them today, but he called them animalcules because they looked like little animals to him. While Leewenhoek was staring at his teeth gunk, he was also sending letters to a scientific colleague in England by the name of Robert Hooke. Hooke was a guy who really loved all aspects of science, so he dabbled in a little bit of everything, including physics, chemistry, and biology. Thus it is Hooke who we can thank for the term the cell, as he was looking at a piece of cork under his microscope, and the little chambers he saw reminded him of cells, or the rooms monks slept in their monasteries. Think college dorm rooms, but without the TVs, computers, and really annoying roommates. Hooke was something of an underappreciated scientist of his day, something he brought upon himself as he made the mistake of locking horns with one of the most famous scientists ever, Sir Isaac Newton. Remember when I said Hooke dabbled in many different fields? Well, after Newton published a groundbreaking book on how planets move due to gravity, Hooke made the claim that Newton had been inspired by Hooke's work in physics. Newton, to say the least, did not like that, which sparked a tense relationship between the two that lasted even after Hooke died, as quite a bit of Hooke's research, as well as his only portrait, was misplaced due to Newton. Much of it was rediscovered, thankfully, after Newton's time, but not his portrait, as sadly no one knows what Robert Hooke looked like. Fast forward to the 1800s, where two German scientists discovered something that today we might find rather obvious, but helped tie together what we now know as the cell theory. The first scientist was Matthias Schleiden, a botanist who liked to study plants under a microscope. From his years of studying different plant species, it finally dawned on him that every single plant he had looked at were all made of cells. At the same time, on the other end of Germany was Theodor Schwann, a scientist who not only studied slides of animal cells under the microscope and got a special type of nerve cell named after him, but also invented rebreathers for firefighters and had a kickin' pair of sideburns. After studying animal cells for a while, he too came to the conclusion that all animals were made of cells. Immediately, he reached out via snail mail, as Twitter had yet to be invented, to other scientists working in the same field, and it was Schleiden who got back to him, and the two started working on the beginnings of the cell theory. A bone of contention arose between them, as for the last part of the cell theory, that cells come from pre-existing cells, Schleiden didn't exactly subscribe to that thought, as he swore cells came from free cell formation, where they just kind of spontaneously crystallized into existence. That's when another scientist, named Rudolf Virchow, stepped in with research showing that cells did come from other cells. Research that was actually, how to put it, borrowed without permission from a Jewish scientist by the name of Robert Remak, which led to two more feuding scientists. Thus, from teeth gunk to torquing off Newton, crystallization to Schwann cells, the cell theory came to be an important part of biology today. Some things we know about science today may seem boring, but how we came to know them is incredibly fascinating. So if something bores you, dig deeper. It's probably got a really weird story behind it somewhere.
There we go. So the type of microscope that you saw was very different in terms of its construction compared to the ones that we're going to be using in class. What I'm doing is, this is what we call a compound microscope. And in this compound microscope, I'm going to ask you to go to your textbook on page 354 and get familiar with the names and the structures of this microscope, particularly how to control, for example, the magnification and the actual objectives with these two particular knobs here, because it's going to be very important in terms of how you prepare slides and how you're able to then view the different structures, or what we're going to say the different either fixed mounts or wet mounts of the cells that we're going to look at. Okay. So in our particular book here, we know that there are two general types of cells. There are the prokaryotic cells and there are the eukaryotic cells. And in general, there are going to be plant and animal cells. And as some of you might know, one of the main differences between the two is that the eukaryotic cells will have the DNA that is going to be bound within a nuclear envelope, whereas a prokaryotic cell has the DNA in a nucleoid region, condensed and still organized, but not separated completely from the cytosol, which is what is going to happen with the eukaryotic cells. Also, you're going to note that the eukaryotic cells will contain many different subcellular structures called organelles. And the organelles are going to be specialized machines, shall we say, in making either glucose, storing toxins, breaking down old cell parts. So these two types of cells are going to be very different. What they will have in common is the plasma membrane. They'll also have DNA. That's another thing that will be in common. They will also have in common with them cytosol. And they will also have in common ribosomes for the sites to where proteins are being manufactured. Granted, the ribosomes of the prokaryotic cells are going to be slightly different than the eukaryotic cells. Their function will still be the same. Another person that I want to introduce you to is Lynn Margulis. She came in this endosymbiotic theory. And if you go ahead and copy that site, you'll get an article in terms of her, as she passed away a couple of years ago in November. She came up with the idea that these smaller prokaryotic cells were literally engulfed by larger prokaryotic cells, pretty much like a large Pac-Man, shall we say, but the membrane that enclosed around them. The difference is that the prokaryotic cells that were engulfed were not digested, but rather the two types of cells began to live in unison, almost what we consider to be a mutualistic relationship. And what we have in terms of the evidence for that are the chloroplasts and the mitochondria. So to give you an idea of what this looked like, the cyanobacteria, very similar to that of the chloroplast in terms of how it's able to capture light energy and make sugars. And then here we have a protobacterium that's very similar to the mitochondria that can break down sugars to make ATP. So these two organelles, the mitochondria and the chloroplast, it's very possible that they were once engulfed by an ancestral host cell. And then what we have now, this is the key, many generations of evolution. These two organelles then became, within the eukaryotic cell, very significant energy transforming organelles. What we have here is some of the evidence to look at that the endosymbiotic theory does occur, is both the mitochondria and the chloroplast, both of them have their own DNA. So they can divide independent of the larger eukaryotic cell. And their own DNA is very similar to that of modern prokaryotic cells. Both the mitochondria and the chloroplast can also manufacture their own proteins because they have their own ribosomes. So the mitochondria and the chloroplast will make proteins significant for their function, whether it is ATP synthase, whether it would be proteins that are found along the thylakoid membrane to capture light energy, or the proteins that are found within the cristae to help with respect to the electron carriers. So these are just a couple of pieces of evidences that will show that the endosymbiotic theory is going to have some support with respect to the mitochondria and the chloroplast having been engulfed eons ago and continue to carry on their own life within our cells, even now. The last thing is that with respect to the structures themselves, rather than going in class and just going over all of these, I want you to construct some sort of data table, whether it's use of numbers, pages, or binder paper, and I want you to make sure that you're going to name the cell structure itself, the structure. It could be the plasma membrane, it can be the mitochondria, it could be the large vacuole, etc. I want you to determine, is this going to be in terms found in a prokaryotic cell? Is it going to be found in a eukaryotic cell, whether it's an animal cell or plant cell, or is it both? So I want you to compare and contrast these different types of cell structures. And in your textbook, go ahead and refer to page 376 for some information in terms of the structure and the function of the cells.